Today we will discuss about matrix rigidity related mechanobiology studies. So what does this means that we will understand about what are the changes that occurs within the cells because of the changes in the stiffness in the extracellular matrix that is surrounding the cells. Though we have covered this topic in our previous videos, we will cover some different aspects of this mechanobiology studies in this video. So we will start with this figure on the left hand side of this panel A. So in this case we can see that when the stiffness of the extracellular matrix changes from soft to stiffer, it might cause different changes, and some of these changes are shown here. Is that when in the case of stiffness, the interactions between the mechanical-biological interactions between ECM and the cells are minimum. However, when the ECM is stiffer, the interactions between ECM and the cells because of the mechanical-biological forces is high, and that might lead to the changes in the nucleus, and at the same time this chromosome got disintegrated. So then the effect of this will cause effect of this will lead to the epigenetic changes or changes in the transcriptions, and that might even leads to this tumor onset metastasis or inflammations. At the same time, it might also cause senescence aging. Senescence aging is, you can understand that cells will continue to survive, however they will not grow. So cells will still remains at the senescence stage, and this could be because of the mechanical-biological forces when the ECM is stiffer. However, when the ECM is not much stiffer, then it might cause this cell spreading, migrations, proliferations, differentiations, which are kind of regular path. And however, when the stiffness of the ECM is changes or it become more stiffer, that it might cause pathophysiological phenomena that we have just discussed. Next, it's a brief overview how these interactions with this matrix rigidity related mechanobiological studies are being studied. Previously, there are studies in 2D conditions that can measure elasticity, cytosol mechanics, single cell behavior in vitro studies, and now there is a shift from these 2D models to the 3D models that can measure dynamic viscoelasticity in nuclear mechanics and collective cells behavior. Instead of just single cell behavior, they can measure the collective cell behaviors, and sometimes one cells might affect the affect the other cells, and because of these mechanobiological forces. So it's a good idea to study collective cells behavior, and the in vivo evidence they can be, and these results about results can be verified using in vivo studies such as in mouse models or humanized mouse models. So these are just a basic overview. Now let us understand the tissues that are found in different parts of the body has how much stiffness. So in the case of this blood fluid, it has a rigidity of around 0.1 kilopascal, whereas for liver it's around 1 kilopascals. Then for skin, lungs, then it will increases for skin, lungs, and by the time it reaches to the muscle, it has a tissue rigidity of 10 kilopascals. And the highest tissue rigidity is for this bone, which is of around 2 to 4 gigapascal. As expected, bone is our hardest material, or is the hardest material of the human. So, and how these studies are being carried out in the 2G engineered model is that we can culture the cells at the center of this petri dish and then observe the migrations or metastasis. After some days, then cells will migrate from this center to this periphery of the petri dish, and these are the cells which are commonly used, such as chondrocytes, cancer cells, neutrophils, osteoclast, and MSC or mesenchymal stem cells. And apart from this, the other studies that can be done is this flexible macropost array. So in this case, The osteoclasts and MSCs are used, and in this flexible micropost array, different kind of different cells are placed at the different spots, and each of these spots, or most of these spots, are varying in terms of stiffness, so that they can measure, so that the person can measure, or the researchers can measure the effect of the cells on different stiffness. And the other things is that in situ softening or stiffening gel. This thing we have covered in our previous videos. You can go through the I button for the link, in which the cells will observe both the soft stiffness and the soft ECM and the stiff ECM, the soft thickness and the stiff thickness, while moving from one sides to other sides. So what is the effect of this is that in the case of this flexible macropost, they can see the behavior of the cells both in the soft stiffness and the both in the soft ECM and the stiff ECM. And the migrations, what the common thing that has been observed is that compared to the stiff ECMs, or stiffer thickness ECM, stiffer rigid ECM has more migrations properties compared to the soft ECM. And when it comes to the differentiations, the soft ECM leads to this adipogenesis, whereas the stiff ECM will cause osteogenesis. And for this in situ softening and stiffening gel, this we have discussed in our the previous videos, it might cause this mechanical memory. That is, when the cells are moving from the uh, soft ECM or soft tissue to the stiffer tissue or stiffer ECM, then the cells might the changes that cells have occurred in the soft ECM will continue to occur in this when the cells are in the stiffer ECM. And similar things has been observed in the reverse case, that is when the cells move from stiffer ECM to the softer ECM. So that has been shown here. So this is just a basic idea, that is about how the matrix rigidity would be related to the mechanobiological studies, and what are the things that are been generally found. If you need to understand more about this matrix rigidity and mechanical biological relations in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.